So on behalf of the co-organizers, myself, Julia, Gerald, and Shaeri, I'd like to welcome you to the second annual Symposium on Infectious Diseases. It's a joint symposium between the Institut Pasteur and QBI. As we were just saying, the goal was to have this symposium several months ago in person in Paris. Of course, we couldn't do that. This is hopefully the next best thing. And the first symposium we had was actually in San Francisco in March 2019. Here's a picture of a number of the participants on the left. This is us in the mission somewhere having fun. And on the right,
I can't see the photo.
Nevan, we don't see your slides.
Oh. Do you see them now?
Yeah.
We see.
Yes.
Oh, okay. Do you see the second slide?
No. No.
Sharing is paused. Bring your
slide.
shared window to the front. What does that mean? That's what it's saying.
What does it say?
Sharing is paused. Bring your shared window to the front.
When you're sharing, do you click on the keynote itself?
Do you see that?
Yes.
Yeah.
Yeah, we always see only the first slide.
With the second slide now?
No?
Yeah,
Oh, yeah, there it is.
Now good. Now good.
Okay, great. So here's a picture of the first symposium on the left of us enjoying a few drinks in the mission. And then on the right is a picture where we're actually discussing in Paris the content of the second symposium. So hopefully we'll be able to get together soon in Paris for the maybe third symposium. But what I'd like to say is we have been working together, scientists at QBI and at the Pasteur Institute after the pandemic hit, working on SARS-CoV-2. And this was a relationship that actually gave a lot of great science. There's a number of papers that are listed that really couldn't have been done without this relationship that was forged before the pandemic. There's a number of media outlets there that highlighted our work. There's a picture of Marco looking good on ABC, and then a number of other media outlets highlighted the relationship between UCSF QBI and the Pasteur Institute. And then on the right here was an article written and put out, I think, by the French consulate describing this particular collaboration. And on our side, this was all done in the context of the QBI Coronavirus Research Group, QCRG. So it's sad we're not together in person, but I'm incredibly proud of the fact that the relationship that we formed before the pandemic led to some really great science over the last several months. Simply put, this work would not have been able to have been done if we didn't have this relationship in place over the last couple of years. Okay, so I'd like to give you the most important slide. I'd like to thank the people that put this together, the QBI media team. As usual, they've done a fantastic job. Jacqueline, Gina, Alexa, and Carolina. And with that, I just want to quickly allude to the agenda. It's over 3 days. We have talks today and tomorrow. And then on Thursday, there's a roundtable discussion amongst the scientists about how further collaborations can go forward, and also a discussion hopefully about potential funding mechanisms to help the collaborations that are already ongoing and the ones that'll be starting hopefully over the next couple of days. And we have just a couple of other people who are going to say a few words as an introduction. First, Christophe Donfer, a professor and scientific director of the Pasteur Institute, then Frederick Young, the new French consul in San Francisco, and then Reid Shabman from NIH, who's a program officer who oversees a large portfolio of grants focused on infectious disease. So maybe I'll just let Christophe now say a few words as an introduction.
Thanks, Nevan. It's really a pleasure to have this meeting today. We were supposed to have a presential meeting scheduled March 16 and 17, and for French people, March 17 sounds like the first day of the confinement. So we had picked a very bad date for our second meeting. But so I'm very happy that eventually we are able to get together and share work not only around COVID-19 and SARS-CoV-2, but also around other pathogens that we are on which we are working at Institut Pasteur in Paris. I think the interaction between QBI and Pasteur started now about 2 years ago when you visited Paris and wanted to actually set up a partnership between our two institutions to try to bring your expertise in quantitative biology and our expertise in infectious disease. And so we've been working on trying to have this partnership. It's not signed yet because the pandemic stopped the administration, but I think the pandemic started a very fruitful collaboration that you have mentioned in your introductory remarks, and especially the collaboration between the group of Marco Vignuzzi and your group and all the groups that you have assembled in this fantastic initiative that has been so productive. So my hope is that from this second meeting, we can start additional collaboration in very different fields of infectious disease that will be as productive as the one that has started thanks to these pandemics. One thing I noted when we discussed just before starting this meeting is that the way science is done has changed a lot with the pandemics. All the collaboration that was done around SARS-CoV-2 and you have led was done without meeting. And I think this is impressive in that sense and is telling us a lot about the future. So I would like to thank your group for organizing this meeting, but also people at Pasteur that have been involved from when we started planning the second meeting, Julia Chamot-Rooke, Carmen Buchrieser, and Gerald Spaeth. And so I wish a very fruitful meeting. I hope that the discussion on Thursday afternoon will allow us to define new projects and go ahead with the partnership. So thank you.
Thank you, Christophe. That's a fantastic introduction. And we're going to segue now to Frederick Young, the Consul General of France in San Francisco, just to say a few words as well. Are you there, Frederick? He was there. Did we lose him? Looks like it. Okay, well, if we can get him back, we'll get him to say a few words.
I'll contact the consulate. Perhaps we go to Reed.
Okay, so Reed. Reed Shaban is also here. He's a program officer from the National Institute of Allergy and Infectious Disease at NIH. And as I said, he oversees a number of large collaborative grants focused on infectious disease. And I just asked him to say a few words about ongoing efforts at NIH and how those efforts could impinge on collaborations like this.
Thanks, Kevin. I'm going to go ahead and share my screen.
Sure.
Apologies, I was cut off.
Oh.
Sorry about that.
Why don't we let Reed speak, Frederick, and I'll let you finish off the intro. Is that okay?
No problem. Absolutely. Thank you.
Okay, great. Thanks. I'll be brief. I think you can see my slides. So thank you for the opportunity to present today. I'm just going to give a couple of remarks about quantitative approaches and host pathogen interactions and countermeasures, ways to identify them from the NIAID perspective. And so the National Institutes of Allergy and Infectious Disease, or NIAID, the mission is to conduct and support basic research and apply it to better understand, treat, and ultimately prevent infectious, immunological, and allergic diseases. And how does this all tie together? So within NIAID, I sit in the Office of Genomics and Advanced Technologies, and OGAT programs drive innovation to accelerate diagnosis, prevention, and treatment of infectious disease. So another way to look at this along the product development pathway is from basic research all the way to clinical evaluation. You would move through the product process, diagnostics, vaccines, and therapeutics. Our office sits more on this basic preclinical development side, where we're looking at supporting programs that are using large data sets and that lead to hypothesis development and testing, which might enable some of these later downstream applications, though it's not completely limited here. And so one specific example that Nevan alluded to is a program that we support, which we call the Systems Biology Consortium for Infectious Diseases. And I'd just like to highlight that this is a community with it that's supported through OGAT that integrates experimental biology, computational tools, and modeling across time and space. And the ultimate goal is to develop strategies that can predict and alleviate disease severity across multiple human pathogens. These groups focus on antimicrobial resistance, tuberculosis, influenza, and emerging infectious diseases. There are 11 of these centers, and they're sort of broken up into two specific groups. So there's systems biology for antibacterial resistance. So these are AMR, focused mostly on AMR. They're in blue here. And then there's also the next generation for infectious disease. This is 5 centers. And these focus on both bacterial and viral pathogens. And to highlight one of these here, obviously today, is the Host-Pathogen Map Initiative, which we'll be hearing more about, obviously, in the next couple of slides. And so this is how they're spread out geographically. And the way that we see this is the benefit of these centers is you can address complex questions across pathogens. And these complementary views can uniquely address omics challenges or large data challenges. So I mentioned the pathogens that are being studied. There's a variety of hosts. I think what's most interesting here is the types of data that's generated. So there's clinical data, genomics, transcriptomics, proteomics, metabolomics, et cetera. These outputs can then translate into global pathogen host networks, predicting vaccine efficacy and patient severity. A number of software and tools can visualize data, perform advanced analytics. Microbiome studies can be applied from this data to harness the patient's gut microbiome to cure infection, and also advancing drug treatments to improve TB outcomes. And this data is put into public repositories for secondary analyses and reuse throughout the communities. And I think the final thing I'd say is, we've had to transition a lot of these large data sets into the SARS to apply them to the SARS-CoV-2 epidemic, correct? And I don't have time to go into all of it, but I'll just point out a couple quick examples. So taking proteomics and transcriptomics studies and applying it to understanding SARS-CoV-2. And here, this Nature paper from the Krogan group and others have shown a protein interaction map, which can reveal targets for drug repurposing. Out in bioarchives from our script centers, look at transcriptomics from patients with mild or severe cases of SARS-CoV-2 to try to get some signatures of what those signatures might look like. And then some other specific examples include system serology. Here's some work that shows distinct early serological signatures from serology that you can track with either survival or antibody responses that might predict convalescence or mortality. And small molecule screening, so setting up large screens to look at antiviral drug repurposing is another way that these centers have taken established platforms and applied it to understanding and ultimately combating SARS-CoV-2. And so with that, I hope that gives you a brief overview of some of the quantitative approaches that that folks have used to identify host pathogen interactions and effective countermeasures. Look forward to hearing the rest of the symposium, and thanks to all the organizers and also to Liliana, our office director. So with that, I will turn it back over.
Thank you, Reed. That's a fantastic summary. We look forward to exploring with you potential funding relationships, creative ones between Americans and maybe the French to help facilitate the type of science you're going to hear over the next couple of days. So thank you very much. And then we'll jump now to Frederick Young, the new Consul General of France in San Francisco, just to wrap up the introduction part of the symposium.
Thank you very much, Nevan. It's interesting to see that even in the heart of the Silicon Valley and San Francisco, you can actually encounter internet connection problems. Thank you very much for having me and for giving me the opportunity to say a few words at the opening of this second edition of your symposium on infectious diseases. I'm very honored. I'm as you said, the new Consul General of France in San Francisco. I just arrived in mid-August, and as a diplomat, my job is actually to contribute to building bridges between, especially our countries. Building bridges in political terms, building bridges in economic terms, cultural terms, but also, of course, scientific terms. And I have in my team two amazing counselors, Jean-Baptiste Borde and Maxime Benalawa, whom we might know, who are in charge of supporting the establishment of such bridges between French scientists and American ones. So this symposium is the perfect example of an amazing cooperation in order to bring closer together two world-leading institutions in research on infectious diseases, QBI and the Pasteur Institute. This seminar is about infectious diseases, but I suspect one of them might get very specific attention. All of you have spent a lot of energy in contributing to the research on COVID-19. Allow me therefore to thank you all personally for the incredible effort and devotion you've shown in your work. One could have expected that in order to combat this pandemic, the political priorities of all nations would be to build international cooperation to support researchers worldwide in a worldwide collaborative and scientific effort to fight the disease. Unfortunately, in the past year, we have to recognize that we've seen also a lot of international division. This is why we should even more so, praise the great cooperation between UCSF and Pasteur. Together, you've already deployed innovative strategies to understand the way the virus uses human cells, and also to identify dozens of potential existing drugs that could be used in order to fight the virus. And you've done this in a very short time. These astonishing results were possible thanks to the trust and respect between the researchers of both your institutes. And you were able to establish this transatlantic cooperation progressively. You started it, as you said, in more quiet times, and this allowed you to react fast and efficiently in this more challenging period. And being far apart, as I actually mentioned during our little chat before the symposium, also gives you a special understanding of the virus. When France was reaching its first peak of infections back in April, the levels in California were still very low, and it allowed pressure sharing of experience and findings. And this flipped around in August when the infection was at its lowest in France, but peaking in California for the first time. Now we are again, unfortunately, in reverse positions, and California is reaching now a 3-month low in the infection, whereas France and Paris are in the midst of a second wave. This just shows how crucial it is to build bridges between your two institutes. So thank you again for your hard work. I wish you a very fruitful symposium, and I hope to see you all very soon in person, maybe at the next symposium. Thank you very much, Nevan. Thank you to all.
Thank you, Frederick, for those remarks, and thanks for everybody for these introductory comments to set the stage over the next 3 days. So with that, I think we'll transition perfectly on time to the science. And Felix Array is going to be chairing the first session. And I just want to remind the speakers that it's 10-minute presentations plus 5 minutes of questions. And at the 8-minute mark of their presentation, there's going to be a red dot that comes up on the screen to let you know there's 2 minutes left.
Okay, great. So,
Felix, go ahead.
Okay, so it's a great honor to chair this first session of this great symposium here. And it's also, I'm very pleased to introduce Olivier Schwartz, our friend and colleague at the Institut Pasteur. Olivier is head of the Virus and Immunity Unit, and he will tell us exciting data about the modulation of SARS-CoV-2 fusion. Olivier, 10 minutes.
Okay, well, I don't know the exact timing of my presentation, so I will try first to share my screen with you. It's here. Can you see it?
Yes.
I will try to use the full screen option. Does it work?
Yeah, just try to go to the second one just to see that it's not there. Great. It's working.
That's
Good.
fine.
Yeah.
Okay.
10 minutes from now, I guess.
10, yeah, 12, okay. Well, no, thanks to the organizers for the invitation. It is a pleasure for me to be here, and I remember pretty well our visit to San Francisco, in March 19. We had such a warm welcome by Nevan, Jacqueline, and all the teams involved. But this was in another life, and nowadays I'm currently with my lab working on SARS-CoV-2 replication, pathogenicity, and host innate immune responses. So today we'd like to share with you some of our recent results regarding SARS-CoV-2 fusion and how the innate immune system may counteract viral spread. I will start by a few slides on SARS coronavirus and its pathogenicity. So in this first slide, you can see the different categories of respiratory viruses. You have the first category of viruses which infect the upper tract and which cause a mild disease, such as seasonal coronaviruses or rhinoviruses, whereas other respiratory viruses like flu or SARS-CoV-2 will reach the lower respiratory tract and will cause much more severe disease. Regarding SARS-CoV-2, there are different cells which are targeted by the virus, in particular in the airways. Within the bronchi, you have airway-ciliated cells, whereas in the bronchioles, you have other kinds of cells, namely type 2 pneumocytes. Why some cells are targeted and the others are not targeted by the virus? It's all about expression of the receptor, which is a molecule termed ACE2. And there are other important molecules that I will discuss, like the serine protease, TMPRSS2, which facilitates infection. So you can see here in this single cell RNA-seq analysis that among pulmonary cells which express ACE2 are mainly type 2 pneumocytes as well as ciliated epithelia. And that's also the case for TMPRSS2. That's why those cells are mainly targeted by the virus, whereas immune cells present in the lungs do not express those receptors and are not productively infected. So that's the role of the cellular proteins to facilitate viral entry. The host immune response is very important, and you know that the type 1 interferon response might be impaired in severe cases of infection. There are multiple articles describing this impairment of type 1 interferon response. I just wanted to quote two of them from Jean-Laurent Casanova's group here at Necker Hospital and at Rockefeller Institute. And he described, for instance, that among patients with life-threatening critical COVID-19 disease, about 10% of those individuals display neutralizing autoantibodies, which will impair type 1 interferon responses and the production of ISG, interferon stimulated genes. That's also the case for other kinds of inborn errors. For instance, the RF7 pathway and TLR3 molecules are also impaired in about 4% of those severe critical cases of COVID-19. So the important question now is, what are the downstream elements or components which control this antiviral response mediated by type 1 interferon? And probably some ISGs, interferon stimulated genes, are controlling, restricting viral infection. And that's precisely what we have been studying in the lab. We have a long-standing interest for ISGs termed interferon-induced transmembrane proteins, IFITEMs. There are 3 human IFITEM proteins, IFITEM 1, 2, and 3. IFITEM 1 accumulates at the plasma membrane, whereas IFITEM 2 and 3 are found within endosomes. And those small proteins, 14 kilodalton, infiltrate the membranes in which they reside, and they will alter the biophysical properties of this membrane. They will increase their rigidity and provide a kind of shell which may prevent against viral fusion. So that's exactly what we have been studying regarding SARS-CoV-2 entry and fusion. So here you have a viral particles which will bind to ACE2, and the serine protease may cleave both, actually, the spike as well as the receptor. So that's what we have been studying. And we were interested not only in infection or fusion mediated by cell-free viral particles, but also what happens once a cell is productively infected, and how this infected cell may fuse with neighboring uninfected cell to spread the infection. And those syncytia, which are formed upon infection, were not precisely described upon SARS-CoV-2 infection, and that's what we have been doing recently. I just wanted to quote an interesting work by Mauro Giacca, describing in severe cases of, in autopsies of patients who died from COVID-19, describing the presence of these large syncytia here, and we know that they produce SARS-CoV-2 antigens. So those cells, those syncytia are productively infected, but there are very interesting questions. What is the role of those syncytia? Are they a kind of dead end for the virus? Are they, on the contrary, a means for the virus to spread more efficiently or to trigger an immune response? So that's exactly the questions that we have been addressing. So I would like to share a few results. We used different cell systems. Here, a few results with U2OS cells, we use those cells because they are highly sensitive to infection when we added the virus, the receptor ACE2. And we also introduced the GFP split system in these cells. So in these cells, you have one half of the cell which will express one part of the GFP protein, whereas the other half of the cells will express the other part. And when the cells will fuse, they will generate a GFP signal. And you can see here fusion, which is generated by upon infection of those cells. We can do real-time imaging of infection in these cells, as shown here, non-infected cells on the left, infected cells on the right panel, and over time, within a few hours of infection, those cells will fuse and produce viral particles. By the way, we term this experimental system the S-Fuse assay because it is highly sensitive, very simple, and in less than 18 hours, we can screen sera, drugs, monoclonal antibodies, and generate this kind of signal. So with this system, we can test, and we are currently testing, again, neutralizing activity of sera, antiviral molecules, and we are titrating also viral stocks and isolating virus from human samples. So what is the role of IFITEMs in this system and TMP-RSS2? So we have generated cells, expressing or not expressing IFITEM. Again, you can see cell fusion occurring very rapidly without IFITEM1, whereas in the presence of IFITEM, there are very much less syncytia which are formed over time. So we can quantify this process, and we have about 4-fold decrease in syncytia formation in the presence of IFITEMs. We also used another cell system in which we only express the spike protein here in 293 T cells, and those cells fuse when they express ACE2, and when TMP-RSS2 is present, there is an increase of syncytia formation. We can also follow in real time the role of spike in cell-cell fusion. And you can see here that there are some algorithms that we can use that measure the GFP positive area among the cell culture. And when we do that in real time, in light gray, you have the syncytia without TMP, and with TMP, there is an acceleration of syncytia formation, which was not known. So the presence of TMP enhances and accelerates cell-cell fusion. And to go fast, what we have shown also is that in this system, IFITM proteins restrict infection, whereas when we add TMP, there is an enhancement of infection, and TMP counteracts the antiviral activity of IFITMs. So you have a kind of balance between cellular proteins like IFITMs, which block fusion, whereas others like TMPs may enhance infection. We are currently analyzing, this is one of my last slides, how does TMP work? Actually, it doesn't degrade IFITMs directly. This was verified by flow cytometry or by Western blots, but it affects the levels of spike at the cell surface, and it cleaves ACE2 molecules. So to conclude, we have shown that SARS-CoV-2 infected cells may form syncytial culture. IFITMs block SARS-CoV-2 fusion. TMPRS2 molecule accelerates fusion and counteracts IFITMs. We are currently performing experiments with primary cells, and I have 2 more slides to share with you regarding the impact of infection on primary human airway cells. And this is a work being done by Lisa Chakrabarty within my unit with other scientists and postdocs. This is a scanning electron microscope view of uninfected epithelial system, whereas when those cells are infected, there is a major damage in the appearance or the motility of the ciliated cells. They lose their cilia, and they are highly sensitive to infection, as shown here. And this is another example, that was my first slide, describing the huge amount of our particles which are produced.
You need to wrap up now, yes.
Okay, and I think that's over. I just wanted to thank the different people involved in this work. Julian Buchreiser, who did some of this work with Jérémy Duflo, Florence Guivel, Mathieu Hubert, Blandine Monel, and Delphine Planas. And sorry for being a little longer than expected. And I don't know if I have time for a few questions. Thank you.
Okay, thank you very much. So maybe we have a few questions. I know Melanie Amon, you have a mic. You can ask your question, Melanie.
Hello, Olivier. Thank you. I had a question, a very general question. So on your first slide, you showed the upper respiratory tract and the lower respiratory tract. If I'm not mistaken, upper respiratory tract epithelial cells also express AC2. So then why would the virus specifically go to the lower respiratory tract? Or is it that we're studying only where it's causing disease, but there could actually be some carriage in the upper respiratory tract which isn't causing symptoms?
That's a very important point. In most cases, you know that the infection will remain asymptomatic, probably because the virus is restricted to the upper respiratory tract. So why would it move? Is it the ability of the virus to spread more efficiently, the occurrence of the inflammatory response, the low level of the type 1 interferon response? Those are probably the different parameters which regulate the access of the virus to the lower respiratory tract.
Thank you.
Okay, so there is a question from Kivan Shokat. He says that he asked if the IFIDM1, which is at the surface, does it have more of a role on controlling syncytia than the others?
That seems to, yeah, that's a good point. That seems to be the case. IFIDM1 is mostly found at the cell surface, but IFIDM2 and 3 transit through the plasma membrane, and we know that that's probably why they are also active, but they are in our hands less active. And you can mutate IFIDM3 by removing endocytic signals. Then IFIDM3 will accumulate at the plasma membrane and will be more active against cell-cell fusion.
I see. So there's another question by Tiziani. What is the effect on the number of cilia?
What is the effect on the number? So that's a work currently being performed by Lisa's team. And there is a rapid disappearance of the number of cilia. In some cells, they lose all their cilia. We don't know what are the underlying mechanisms. It is, Lisa is currently studying which transcription factor, for instance, regulates the tubulin network, which is important for the formation of these ciliated protrusions.
Okay, so another question is, which kind of fusion inhibitors are you testing in your fusion assay? I guess other than the FEDMs.
We are testing different kinds of molecules. Well, first we are looking at the effect of monoclonal antibodies, which bind to the spike, which neutralize the virus. And we know that they are able to neutralize as well cell-cell fusion. This is a work currently being done by, in collaboration with Hugo Mouquet here. And we are testing a few molecules from people who are interested in studying the formation of syncytia. We have also performed a screening of other ISGs, which may impact syncytia formation, but this is ongoing work.
Okay, well, thanks a lot, Olivier. Very interesting talk. Thank you. So we'll move to the next presentation. It is Jeff Cox, who's a professor in the Department of Biochemistry and Molecular Endocrinology, and also at the Department of Immunology and Pathogenesis at UC-SF, and is a member of QBI. And the title of Jeff's talk will be Host-Pathogen Interactions using Global Proteomics to Discover New Anti-TB Pathways of Macrophages. So Jeff, please go ahead.
Thank you, Felix, for that introduction. I hope, I just want to make sure that everyone can hear me, Felix.
Yes, everyone can hear you.
Great, fantastic. Okay, great, fantastic. It's my pleasure to, I'm sorry?
Just try moving to the next slide to make sure it's working.
Here we go.
Yeah, perfect.
I'm gonna go back. It's my pleasure to be able to share our work with you all today, and my colleagues, especially at the Pasteur. And my lab studies an organism that also causes a pandemic. In this case, it's a pandemic that's preceded SARS-CoV-2 and will likely hang on after COVID-2 goes away, hopefully. Well, hopefully COVID-2 goes away. To cut right to the chase, TB is an intracellular pathogen. It's a Mycobacterium tuberculosis, and it lives inside macrophages. When it gets inside macrophages, it's thought to maintain itself inside a vacuole, but that we know from work from my lab as well as from my colleagues at the Pasteur, Roland Broche and Jos Eninga, that TB pokes holes in that membrane. And it's that simple act of poking a hole in the membrane, access to the cytosol, that changes not only the biology of the bacteria, but also changes the biology of the host cell, of the macrophage. And that's something that my lab has been interested in quite some time. And there is a secretion system from TB called the type 7 secretion system, or ESX1, that is responsible for mediating that membrane perforation that then changes this dynamic between host and pathogen. What I'm going to be talking about today is really one of the reactions that occurs in response to membrane permeabilization, and that's a process called autophagy. And many of you are likely aware of there are many intracellular pathogens that break down phagosomal membranes, including Listeria monocytogenes, which of course has been studied for many years by Pascal Cossard at the Pasteur, as well as Dan Portnoy at UC-Berkeley, my institute. And Listeria patently gets out of the phagosome and gets into the cytosol and actually moves around inside those cells using actin-based motility. In the case of TB is that we think that perforation is much more limited in that it creates a dynamic between membrane repair and membrane permeabilization. EM studies, most EM studies show that TB stays inside at least a membrane-bound phagosome, but we think that there is, well, we know that there is exchange between luminal contents and the cytosol. And as I mentioned, there are effects that happen when TB pokes holes in the membrane. One of them is the induction of type 1 interferon, which of course is an antiviral response, but it turns out that that also promotes bacterial infection. But what I'm going to talk about today is the role of the ubiquitin-mediated autophagy pathway. And you can see here is that this is a macrophage that has been infected by an M. cherry containing MTB. This is wild type TB that's poking holes in membranes, and you can see that there's a massive amount of co-localization with ubiquitin, and it's that ubiquitin that then leads to the development of an autophagosome, which will eventually deliver the bacteria to the lysosome, and I'll describe that later in the next slide. ESX1 mutants don't do that. They don't poke holes in membranes, there is no exchange, and there's no recognition that then triggers this autophagy pathway. Okay, so here's a summary of what we think, at least our view currently, of how TB interacts with macrophages, in particular with the autophagy pathway. Early after phagocytosis, the activity of ESX1 leads to perforation of the phagosome, and this is counteracted by the escort machinery. This is work that has been done by Jennifer Phillips and others. But the recognition of these membrane pores in the phagosome leads to the ubiquitination of not only the bacteria, but also of the phagosomal membranes as well. And there's a number of ubiquitin ligases, some of which we've been studying and others as well. But if autophagy doesn't continue, then this will turn on, as I mentioned, not only type 1 interferon, but also lead to other effects on the host cell, including activation of inflammasome, cell death, and apoptosis. But a key molecule or reaction that links this ubiquitin target to the autophagy pathway are these molecular receptors called autophagy receptors. And they bind to two different proteins at the same time. They bind to ubiquitin, but then they also bind to an autophagy protein called LC3. Probably many of you have heard of P62, which is a common autophagy adapter receptor that works on lots of different autophagy processes, including and tuberculosis, and localizes to the bug during infection. Work from Ivan Dikic's lab has shown that a kinase, TBK1, phosphorylates P62 after initial recognition, which then leads to a cascade of further recruitment of these autophagy adapters, as well as increased ubiquitination. Finally, this whole gamish of both ubiquitinated membranes as well as phosphorylated autophagy receptors leads now to the production of what's termed the phagophore, which eventually will close and then lead to delivery of the phagosomal contents to the lysosome for degradation. So that's clearly an important part of this whole process, but also too is that the continuation of this process is also important for dampening type 1 interferon responses. So what happens then in cells that can't do autophagy during TB infection? So I'm going to show you a movie here that I want to just very briefly introduce, and that I'm going to show, there's going to be two panels. This is live imaging of bone marrow-derived macrophages from mice infected with wild-type TB, all done in the BSL-3. The bugs will be labeled, the bacteria will be labeled with GFP, and it'll be bright field images of macrophages moving around inside the cell, on the dish. This is work done by a Frenchman in my lab, Guillaume Golovkin, and you're going to see two panels. On the left side will be wild-type macrophages. On the right side will be macrophages that cannot perform autophagy. They're ATG5 knockouts. And the first movie, I'm just going to let it run, and you'll see that there's a lot of cell death. But then the second run of the movie, there'll be little circles that'll come up to show you some interesting things that we can see by doing this kind of live video microscopy. So this first movie, and you can see initially this is over 4 days of infection. And you see that there's a lot of movement of the macrophages, but you can see even after 1 day that now there's a lot more cell death in the ATG5 knockout, these are autophagy deficient macrophages, than in the wild type. You can still see that there's plenty of growth because TB does grow inside these macrophages. But clearly by the end of this experiment, there's just massive amounts of cell death. So again, this is that same movie, but by closer examination, we can see that there's a lot of differences that occur in these macrophages. In that, one is that there's not only early cell death, but there's a process called efferocytosis. These macrophages eat each other, especially one will die very quickly, be very rapidly eaten by another macrophage, and then that macrophage will rapidly die. And then surprisingly is that the bacteria will actually grow inside these dead cells. Okay, so obviously protein post-translational modifications play a big role in targeting bacteria to the autophagy pathway. And so in collaboration with Nevan Krogan's group, in particular with Danielle Sweeney, who you'll hear from tomorrow, is that we looked for differences in post-translational modifications globally inside macrophages that are infected with TB. I don't have time to tell you all the details of this, but of course, by using mass spectrometry, we can identify lots of different changes that occur during infection. And one thing that we've been looking at are these autophagy adapters. I mentioned P62 as a major adapter, but there are a number of other putative adapters, and some of which are known for other bacteria, such as optoneurine, that are involved in this pathway. The one that I want to focus on today is this unique one called Tax1BP1. It has one phosphorylation site that was significantly changed and upregulated during infection, but there are multiple other phosphorylation sites in this protein. So what is Tax1BP1? Just like P62, it has the important domains. It binds to LC3, that's this LC3 interaction domain, the Lear domain, and it has a ubiquitin binding domain here, an interesting one, and it also has a dimerization domain, this thing Multimerase is. This is very similar to P62, which also has this Lear domain, a ubiquitin binding domain, and a dimerization domain. The one unique aspect of Tax1BP1 is that it also binds to myosin 6, and this is work that was primarily done by Paola Arimondo group at the MRC. And that'll become important in just a second. So what happens then in macrophages that lack Tax1BP1? Do they look like what happens in ATG deficient macrophages? And the answer is no. We got a surprise here. And so this is very similar experiment to what I mentioned previously, is that we infected cells with red Mycobacterium tuberculosis and then co-localized ubiquitin using antibodies. And you can see with wild type is that not every bug gets localized. About 25 to 30% of the bacteria normally localized to ubiquitin. When we knocked out the adapter P62, that amount of ubiquitin actually went down, indicating that P62 works early in conjunction with the ubiquitin ligases that lead to deposition of ubiquitin around the bug. But in Tax1BP1 macrophages, we got a very big surprise, and that is that we saw an accumulation of ubiquitin around the bug, indicating that this is blocked in a later stage of autophagosome development than P62 mutants. These macrophages also induce more type 1 interferon, as well as more prone to cell death. And importantly, what I've shown here is that the bacteria actually grow better inside Tax1BP1 macrophages.
I'm sorry, Spaeth, you need to wrap up soon because we're at 10 minutes already.
This is my last slide. And so this is what we think is what's going on, is that we've, although it's still, the Tax1BP1 is very interesting. P62, we think, comes early and is required for full ubiquitination, but Tax1BP1 works later. And so that these autophagy receptors act in sequence. And Tax1BP1 is probably dragging something to the phagosomal membrane to now allow for the completion of the early autophagosome into a completed autophagosome that can then be degraded. So with that, I will end and thank my collaborators, Nevan's group in particular, Danielle, who was important for this. This guy here is John Budzik, who did all the work on the Tax1BP1, and these are our collaborators, and I think to the NIH for funding for this project. So with that, I will end and take any questions.
Okay, thank you very much. Please type your questions on the chat box, and I'll call them to Jeff.
There's a question in the participant box, Felix.
I don't see it.
Yeah, I see it from Joanne.
Yeah, okay, go ahead. You see it.
Yeah. So what happens if we mutate the Tax1BP1 myosin 6 binding site? That's a key question, and we're trying to do that. Unfortunately, that binding site is overlapping with the ubiquitin binding site. I didn't really have a chance to go into that. But when we try to make mutants that don't, we tried the opposite experiment where we tried to make mutants that we thought would not bind to ubiquitin, and it turns out that those also don't bind to myosin 6. And so we're still trying to figure out exactly what the right mutations are to do that. But I think that's a key question. We know that at least from the BUS lab, where they've looked in Salmonella, is that when they knock down myosin 6, that then they see a decrease in autophagy targeting with Salmonella, which is of course another intracellular bacterial pathogen that also utilizes this same pathway.
So can you speak about the possibility that this pathway is involved with other bacteria?
It's absolutely going to be involved in other pathogens. And again, Paola Arimondo group really showed nicely that this is important for Salmonella infection. Whether it's involved in, and it's also involved in other autophagy-dependent mechanisms of targeting, say, like damaged mitochondria to the autophagy pathway. And so I think that this is going to have likely have broad effects on lots of different intracellular pathogens. I think the important point here is that it's not a complete null, that the taxon BP1 is probably an efficiency thing, and that we think that it's allowing for maybe more rapid deposition of autophagy components around the bug. When we knock it out, it's not like the bugs are as severely deficient as like an autophagy deficient macrophage, but that this plays a role in making it more efficient to allow for targeting to happen much more rapidly during infection.
And beyond bacteria, you think other pathogens can use?
I'm sorry?
Beyond bacteria, other pathogens like intracellular pathogens.
Yeah, right, exactly. Salmonella for sure. That's been shown.
Beyond bacteria, I mean, like
beyond bacteria. Oh yeah, well, viruses. I don't think that that's been tested, but we can certainly do that. I mean, certainly many viruses are. Viruses have this interesting interaction with the autophagy pathway. Some of them seem to be sensitive to it. Other ones need it in order for replication to occur. So it'd actually be really interesting to test the role of taxon BP1 and these, two kind of classes of viruses.
So there's a final question. Is there any connections to vacuolar pH?
Is there any connection to vacuolar pH? Yeah, that's a really interesting question. It's hard to actually measure because this process, that's a really interesting question. It's very interesting because it turns out that not every phagosome is the same and so it's very difficult to measure the pH of individual vacuoles. David Russell has tried and you can do it somewhat, but it is very difficult to tell. And so it's an interesting question. We just don't know the answer to that. It's certainly that in the absence of autophagy, and likely then with taxon BP1, is that now there isn't the same amount of acidification, that's for sure. Overall, when you look at them in bulk population.
I see. So I think we need to move on now. So you still have another question. Could you answer it on the chat as we go on? So, okay, thank you. So the next speaker is Paola Arimondo from Pasteur. Paola is head of the Department of Structural Biology and Chemistry, and her title is Why Use Chemical Tools? So design of an epigenetic chemical toolbox to reprogram cells and its applications from cancer to malaria. Paola, please go ahead.
All right, so let's start. So before we start, I want to say I was one of the fortunate ones that was lucky enough to go to San Francisco last time and to be wined and dined, and it was a wonderful experience, and we're very sad that we couldn't host you this time around, so hopefully soon. But in the meantime, this is wonderful interaction. So I'd like to start by the session by presenting Joanne Engel, who will give a first talk. She is a professor in the Department of Medicine and director of the Microbial Pathogenesis and Host Defense Program. And her talk is on chlamydia, which is a master cell biologist. So Joanne, you have 10 minutes, and the red dot will indicate 2 minutes from the end of your talk. Go ahead.
Thank you. You can hear me okay, and you can see my slides?
I can, absolutely.
Great. All right. Well, great. Thanks so much. I'm delighted to be here to tell you about chlamydia as a master cell biologist and the secret life of an intracellular pathogen as revealed by proteomics and other cool things. All you need to know for today's talk is chlamydia trachomatis, which is amongst other things a leading cause of sexually transmitted genital disease, is an obligate intracellular bacteria, and it replicates inside a membrane-bound compartment, so-called vacuole, or in the chlamydia world, we like to call it an inclusion, in order to survive inside the host cell. To do so, it must exquisitely regulate host cell trafficking and organelles and selectively interact with some while not interacting with others. So the question is, how does a pathogen that has a reduced genome of only 1,000 kb establish such a unique niche with only about 900 genes? And it does so in a manner similar to what the Legionella does, which it has a specialized protein export system, the so-called type 3 secretion nanosyringe needle that allows the export of about 100 to 150 secreted effectors across the bacterial membranes and across this inclusion membrane into the host cell cytosol, where they can, of course, interact with host cell proteins and organelles. What's particularly interested us over the last number of years is a subset of these type 3 secreted proteins, the so-called inclusion membrane proteins, or INCs, that are unique to the order Chlamydialis. They too go out through the type 3 secretion system through some kind of topological magic, and then are inserted into the host cell, into the inclusion membrane. They're characterized by having 2 long transmembrane regions connected by a short cytosolic linker, and then their N and their C termini appear to be sort of hanging out into this host cell cytosol, saying, bind to me. The business end in general seems to be at the C terminus, the secretion signals at the N terminus, but bioinformatics has revealed very little into their function. They don't have any obvious homology with each other or to other known proteins or protein domains or enzymatic activities. And because genetics in chlamydia has been quite difficult to do, it's been hard to associate these INCs with specific functions. But we would posit that just based on their localization at the host pathogen interface, that they're ideally poised to interact with host cell components and to form novel scaffolds. And just to set the stage, you could imagine that by recruiting host proteins and native interactions, they may have antagonistic functions on the host landscape, or they could assemble new complexes on the inclusion membrane, perhaps to enhance host functions or to relocate host functions. So in the absence of high throughput, easily accessible genetics, we turn to the next best, which is performing what I like to call a Krogan, which is that we teamed up with Nevan Krogan's amazing proteomics and bioinformatics team and used high throughput affinity purification mass spectroscopy to identify the INC host interactome. And we did this in 2 ways. Initially, what we did was we did what we call our transfection interactome, where we took each of the individual INCs from Chlamydia trachomatis, strep tagged them, transfected them into cells, and then it did affinity purification, mass spectroscopy, and bioinformatics. And from that, we got what we like to call our transfection interactome. In addition, more recently, in work carried out by Ellie McMahon, Jess Shaeri, and Sherry Elwell, we've now done what we like to call our infection interactome. And that is that we've either constructed or obtained from other strains of chlamydia, each expressing one of the INCs with an epitope tag. We then go on to infect these cells, again, carry out affinity purification mass spectroscopy, and develop from that what we like to call our infection interactome. And while we're still in the process of completing this interactome for the about 15 or so INCs that we've looked at, there's actually very good correlation between the infection interactome and transfection interactome, which is quite a reassuring and nice result. So what does this look like? This is what I like to call my typical Tinker Toy diagram. This was based on the transfection interactome. The INCs are in these blue and gray circles, and the spokes go out to the predicted high confidence host protein interactions. I just want to give a call out to Robin Kaki's talk tomorrow. She'll be talking about this INC and its interaction with all 11 subunits of dynactin, and talking about her very cool approach to cross-linking mass spec to identify the direct interactor with CT192. Today, however, I'm going to limit my talk to some really interesting things we found about this interaction. And this interaction is INC-E, an early expressed INC of unknown function. And what both our transfection and infection interactome revealed is it appears to be interacting with high confidence with 2 disparate and distinct sets of host proteins. One are sorting nexins, which were associated with retromer-dependent trafficking, and the other are syntaxins, which were involved in membrane fusion. So let me tell you first about the interaction of INC-E with sorting nexins. Most of this work is published, so I'm just going to give you a couple highlights today. First, let me remind you that what the retromer does is it recruits and selectively concentrates host cargo, such as the mannose-6-phosphate receptor from late endosomes, leads to membrane tubulation, and then the delivery or retrograde transcription of these cargo back to the Golgi, where they typically pick up their usually associated proteins, such as lysosomal hydrolases, which are then delivered back to the late endosome and to the lysosome. And the retromer is composed of 2 subcomplexes, a so-called VPS complex that was previously thought to be primarily responsible for cargo recruitment, and then a heterodimer of either sorting nexin 1 or 2 in conjunction with sorting nexin 5 or 6. These SNCSs are highly homologous, and they have bar domains that are involved in membrane bending, tubulation, as well as dimerization, and a phosphoinositide homology domain, or PX domain, that's involved in phospholipid recognition and membrane binding. And what we found was this INC-E, and specifically just the C-terminal 24 amino acids, was necessary and sufficient to bind with high affinity to sorting nexins 5 and 6. They bring along SNCs 1 and 2 for the ride. They don't bring the VPS complex. And in doing so, they disrupt retromer-dependent trafficking. Together with Oren Rosenberg, we solved the crystal structure of the C-terminus of INC-E bound to the PX domain of SNCs5. And what we find is that this INC-E peptide binds to a highly conserved hydrophobic groove with high affinity interaction, suggesting that INC-E might be disrupting a previously unsuspected native interaction between SNCs5 and 6 and a host protein. So we then returned to proteomics, again in collaboration with Nevan's lab, and I think came up with a method, and really give credit to Oren for thinking of this, that allows us more generally to interrogate then how bacterial effectors might be disrupting host-native interactions. So in this case, we basically determine the interactome of SNCs5, and of course we know that it will bind with SNCs1, and we also found that it interacted with the cation-independent mannose-6-phosphate receptor. We then titrate in the C-terminal INC peptide, and we see now that we specifically disrupt the association with the mannose-6-phosphate receptor with SNCs5 without disrupting SNCs1. And that suggested to us that what INC-E is doing is it's disrupting at least one cargo recognized by SNCs5, the mannose-6-phosphate receptor. So just to summarize this part then, INC-E subverts retromer-dependent trafficking to survive intracellularly. It does so by displacing previously unidentified SNCs5 interactors, such as the mannose-6-phosphate receptor, disrupting retromer trafficking. And we also learned by depleting cells of SNCs5 and SNCs6 that the retromer seems to function, surprisingly, as a restriction factor, which is to say when we deplete cells of SNCs5 and SNCs6, we actually see an increase in the production of infectious progeny. So this story itself was already quite interesting to us, but that's not the whole story. And I want to share you now with, very quickly, just a little of new unpublished data about the other complex that INC-E was thought to interact with, which is syntaxins, involved, as of course you all know, in membrane fusion. And what we found was that INC-E was—
you're going to have to make it very short. Sorry.
Yep, okay. Was predicted to interact with STIX12 associated with early endosomes, and STIX7 late endosomes, could either be doing so by recruiting those proteins directly or by recruiting the vesicles themselves. And as you can see in this spinning disk confocal time-lapse microscopy, you can actually, I think, see evidence that these SNCs7 and SNCs12 vesicles are very close to the inclusion outlined in red, and perhaps maybe even fusion. We also can show that STIX7 is required for intracellular growth, and thus what we'd like to end with is just saying that INC-E functions as a master multitasker to promote chlamydia intracellular survival by bringing these vesicles to the inclusion. It promotes replication, perhaps by bringing nutrients, cholesterol, membranes, and proteins. At the same time, you're disrupting the retromer, which is important for lysosomal fusion, and that's perhaps some kind of a backup mechanism. So if lysosomes do fuse with the inclusion, they might be less effective. And then just to say that intracellular pathogens are master cell biologists, I think this kind of global mapping is broad applicably to genetically challenging pathogens. I think these inks in general are going to function as protein scaffolds. And to repeat what Shaeri Mukherjee has said, dissecting these host pathogen interactions leads to new insights in cell biology. With that, I just want to acknowledge a lot of people from my lab have done a lot of work on this. Great collaboration with Oren Rosenberg. Of course, we couldn't have done this without Nevan's proteomics, bioinformatics, and help from other people in the community. And I'll stop there. Thanks for the extra minute, and be happy to answer questions.
Thank you very much, Joanne, for a great talk. We have a question from Kevin, who's asking, who says the inks look like they have roughly the same size and topology as Olivier's IFIT-M proteins, and they're in internal membranes serving as restriction factors for pathogens. So is there any possibility that they're related?
Great question, Kevan. I'll have to think about that. Hadn't thought about that before.
I'll look in the question and answer.
Melanie, there's a comment from
Yeah.
Kevin, and I see this too, that I don't know if I can't type in questions in the Q&A box for whatever reason.
Aha, yes, I'm not quite
the
sure.
Participants can as well, so that would obviously make it
Okay.
hard.
So then maybe can the participants type in the chat? Or only the panelists type in the chat?
I think only the panelists can. I don't know, I'm just wondering, can the attendees actually use the Q&A box? Because I can't, and I don't think Kevon can.
I think attendees can, but panelists can't maybe.
Got it, okay.
Someone is testing and it seems to work.
Got it, okay, just double checking.
Okay.
Thank you.
Okay, great. I have a question, Joanne. Any of these proteins so the bacterial proteins, do any of them look like eukaryotic proteins or could have homologs?
Right, great question. So in general, the bioinformatics has revealed very few clues of what these do. So in general, they don't have homology to eukaryotic proteins or typical motifs, let's say like by sarcomology domains or enzymatic activities. The one thing we see is sometimes that they're just small coiled regions in the C-terminus, which again would be consistent with them functioning as scaffolds and mediating protein-protein interactions.
Okay, so they've evolved a life of their own. They haven't mimicked anything that actually exists.
As far as we can tell, but there are some that have some motifs, but it's been less, in general, not true. We're looking at several right now that have coiled domains, for example, and that seems to be the key to the interaction.
All right. Let's see if there are any other questions. I don't see any other questions, but we have to move on anyway. So if you have any other questions for Joanne, you can type them in the Q&A. Something just showed up, no test. Or you can email her directly, maybe. So Carmen, thank you very much. We will move on to our next speaker from UCSF, who is Judy Sakanari, and she is professor in the Department of Pharmaceutical Chemistry. And she will tell us about discovering new drugs to treat two neglected tropical diseases, river blindness and lymphatic filariasis. So we see your slides, but we cannot hear you.
So thank you very much for the invitation to talk. I will be talking about our work to identify drugs to treat two neglected tropical diseases known as river blindness and lymphatic filariasis. Let's see here. As you can see, these worms are very long and thin. They're very active, and sometimes they just pose a challenge to our screening. But first I want to talk about the two diseases. The first disease is onchocerciasis, or river blindness. This is caused by a filarid nematode called Onchocerca volvulus. The adult worms are found within the subcutaneous nodules, and then when they're extracted, several adult worms can be found. And these worms are anywhere from 30 to 50 centimeters in length, and they can live from 10 to 12 years. The female worms release these very young stages called the microfilari. And as they migrate through the skin, they cause severe itchiness. And when they migrate up to the eye, it can cause blindness. And as you can see here, this young boy is leading a blinded man by a stick. With lymphatic filariasis, it's also called elephantiasis. It's caused by several species of filarid nematodes again. The adult worms are found in the lymphatics, and they are the cause of the extreme enlargement of the extremities. Adult worms do release microfilari into the bloodstream, but it's really the adult worms that cause the pathology. So together, these parasitic infections cause millions of disabilities. It's a chronic disease in millions of people in mostly impoverished countries. There are drugs that are used in mass drug distribution programs, but they all target the microfilarial or the very young stage, and they do not kill the adult worms. So our goal is to try to identify macrofilaricides, or drugs to try to eliminate the worms and thereby prevent transmission, because these worms transmit millions and millions of these microfilariae over a lifetime. So the way we screen our worms is using what we call the Worminator. This was developed by Chris Marcelino in my lab, who designed a software program that uses a video camera that Chelsea is demonstrating here. The camera looks up at the 24-well plate that contains these individual female worms. As you can see from these plates, we can do IC50s. We do them in replicates from high concentration to low concentration. And the computer software program detects the pixels displaced per second for individual worms, and it calculates the mean movement units. And this is how we're able to actually calculate these IC50s. Again, this is very inexpensive. Less than $3,000 US dollars, and the software is available and it's free. So also you can see here we've repurposed an old Kodak box for our purposes. I'm showing this slide because, as many of you know, with infectious agents, they're very difficult to obtain. So I just wanted to show you the different sources. Fortunately, we can get our Brucia worms from laboratory rodents called GERDs, but our colleagues in West Africa collect the adult worms of Onchocerca from cow hides from the abattoirs. And some of the filarial nematodes are also from humans, and then we also collect them from black flies that are infected that get to feed on humans. One individual worm for Brucia costs $5 US dollars. We use different approaches, and we used repurposed libraries for our screening purposes. These are known drugs with adequate clinical safety and PK information, so these can decrease the cost and time associated with a lot of these preclinical assays. We identified 2 repurposed drugs, Oronafin and Pervinium pamoate. Both of these have been used for decades on humans, and they look very promising as drugs for filarial diseases. Another approach is to identify antibiotics to target the bacterial endosymbiont called Wolbachia. And in clinical trials, patients were treated with doxycycline, and when the worms were removed from the nodules, it was found that the doxycycline actually depleted the Wolbachia within those worms. So Wolbachia, again, are these intracellular endosymbiotic bacteria that the worms depend on for their nutrients. They're found in many, many species of insects, but most filarial worms also have Wolbachia. So doxycycline is effective in affecting the female worm fecundity, reducing their reproduction, as well as their survival, but it takes 18 months of 100 to 200 mg per day for 6 weeks. Doxycycline is also contraindicated for pregnant or breastfeeding women and young children, so these factors really preclude its use in mass drug administration. This image is just showing in red where the Wolbachia are found within the microfilaria itself. The Wolbachia are transmitted vertically from the female worm to the microfilaria. So some of the things that we're concerned about are, because there are major campaigns that are ongoing to identify new antibiotics for these filarial diseases, is what happens if the drugs can eliminate over 99% of the Wolbachia. What happens to that 0.1%? Do they come back, or can that actually eliminate the infection? The other question we have is, of course, deals with antibiotic resistance and whether some of these anti-Wolbachia drugs will cause resistance. So Emma Gunderson and Ian Vogel in my lab wanted to do a study with rifampicin. Most of the studies that have been done in animals with these antibiotics have only been very short-term, limited time points, up to maybe 4 months. And so what they wanted to do was to see the effects of rifampicin on these infected gerds after different time points and up to eight months. So at each time point, qPCR was conducted to look at the Wolbachia titers in the worms, embryograms, and then we did some sequencing to look at late and early populations and confocal microscopy. The bottom line is that the Wolbachia titers did rebound over time. They started out initially eliminated, but they did come back after 8 months. The same with embryogenesis, which is the development of these larvae. And then the sequencing of the Wolbachia genome showed no genetic changes that could account for the rebound. Now, Laura Chappelle and Bill Sullivan's lab found something very interesting when she did the confocal microscopy on these ovarian cells in the worms and found these interesting clusters of Wolbachia. And so what she wanted to do was to ask the question, if you look at the Wolbachia inside the clusters and compare them to the number of Wolbachia outside the clusters within these ovaries, is there a difference between the controls and the treated worms? And indeed, at 6 weeks, Wolbachia surrounding the clusters were reduced in treated worms, but later repopulated the ovaries. So she also found that there wasn't any change within the clusters. So what we think may be happening is that these clusters may be these privileged sites or antibiotic resistance. So just to recap our study, Wolbachia rebounded with a corresponding return of embryogenesis and fecundity after that 8-month period. And interestingly, these clusters have never been reported in these filarial worms, and we are going to pursue more studies on these interesting Wolbachia within the clusters. So with that, I want to thank the fantastic parasite team from UC San Francisco and a number of our collaborators and our pharmaceutical companies that have worked with us, also the funding agency, as well as the organizers. So thank you. And I'm happy to take questions now.
Thank you very much. So for now, there are no questions. Yes, there's a question from Jeff Cox. Love it. Are the Wolbachia in the clusters non-dividing?
That's a great question, and that's something we want to do. So we're thinking of various ideas on how to look at that. We have done fish analysis, so we do know that these Wolbachia within the clusters are living. We'll probably try some BRDU analyses and see if they're actually replicating or not. But yeah, that's a really great question. Then we want to be able to follow their activity within the ovaries.
There's another question from Melanie Ott. Beautiful talk, Judy. Any interactions with mosquito program when targeting Wolbachia?
Yeah. So there's been a lot of interesting work from Scott McNeil's group several years ago when he found out that the Wolbachia had interfered with the dengue replication in mosquitoes. And so that seems to be working out pretty well in Australia when they did the release. So that's very promising. With the worms, just to get back to the filarial worms, they're very difficult to work with because we don't have any nematode cell line that we can actually work with the Wolbachia in culture. So everything is done ex vivo. Thank you for the question, though.
Are there any talks? I have a quick question regarding your screen that you do. So if I understood the method that you're acquiring is you're actually looking for movement in the wells, in your 24 well plates. So then how would you just define as if they stop moving as a drug that is active, but then do you go back and check whether the worms are actually dead or are they just non-motile?
Right, that's a great question. We always get asked whether the worms actually recover when we say they're dead, but when we say they're dead, they really are dead. Morphologically, they become opaque. They definitely stop moving. There are drugs that do paralyze the worms, like levamisole, for example, and the worms do recover. But in our assays, and we screen, you know, tens of thousands of compounds, and found that when the worms actually stop moving, they're pretty dead. We do make a cutoff in our screening at 70%, and then we'll repeat it again and make sure that those drugs, it's just not a one-off, but the hits are really genuine.
Right. All right. Okay, well, it seems like there are no other questions, and we are perfectly on time. So thank you very much, Judy.
Thank you.
Our next speaker is Germano Cecere, who leads the G5 in the Department of Developmental and Stem Cell Biology. And he will tell us about the role of small RNAs during SARS-CoV-2 infection. Thanks. Our next speaker is Nevan from UCSF. He's a professor and vice chair in the Department of Cellular and Molecular Pharmacology, and he's professor in the Department of Chemistry at UC-Berkeley. Also our Howard Hughes Medical Institute. And he will be telling us about leveraging new insights from cancer drug discovery to target viral host factors. So Nevan, the floor is yours.
Okay, thank you, Melanie. Do you see my slide?
I do see your slide.
Great. Okay, thank you for the introduction, and thank you very much for inviting me to the symposium. And I, of course, also wish we could have all been together. We are primarily an oncology drug discovery lab, and I, through interactions with Nevan in March, became very, very interested in how we could repurpose some of our assays and insights to work on this virus. So one common bottleneck for both the immune system to see virally infected cells as well as cancer cells is the presentation of, in the case of cancer, tumor neoantigens. These are point mutations in normal proteins that get recognized as non-self, and then CD8 T cells can attack the cancer cells in a very specific way. That's been a breakthrough in immune oncology in the last 15 years. But of course, viruses, those proteins are much more divergent from self. So if they're presented, they'll mount an even bigger response. But we've heard a lot of great talks about the interferon response, but this kind of adaptive immunity is also very powerful, but viruses are well recognized as having very many tools to block the presentation in the class 1 and class 2 MHC system. And on this panel, you see the many viral proteins from different viruses that intersect at all of the different steps along the pathway of getting a folded protein processed in the proteasome, loaded to the ER, and then chaperoned and bound to class 1 MHC and recycled. So we have been looking at this in the cancer context. We've been looking for drugs that would amplify the specific presentation of cancer neoantigens. And so we wondered if we could repurpose this for the SARS-CoV-2 project. And so Ziyang Zhang in the lab had already developed an assay to look for cell surface MHC presentation of different cancer neoantigens. So he repurposed the assay with David Gordon's clones of the SARS-CoV-2 genes, expressed each of the ORFs in HUK293 T cells, and measured class 1 levels on the surface. And by going through all of the SARS ORFs, we found a number that do suppress the class 1 MHC on the cell surface very strongly. NSP1, 14, and 15, as well as ORF8 were ones that we tended to focus on. NSP14 is what I'll focus on the most. That is the largest magnitude effect. And so we went back then to the protein-protein interaction map from the QCRG, And you can see that now we know which host factors are interacting with each of the viral factors that we identified as downregulating class 1 MHC. NSP1 binds the ribosome, suppresses translation, suppresses host interferon response. NSP14 is a dual enzyme, 3', 5' exo proofreading, and N7 methyltransferase. NSP15 is a uridine-specific nuclease, also host interferon response related. ORF8 is also a type 1 interferon antagonist. So we looked at this and we said, well, NSP14 has the largest magnitude effect at reducing class 1 MHC, and it's not been linked to interferon response or the immune response. So we decided to focus on that. So I showed you the cell surface facts, but now we wanted to know in Western blots whether we would reduce class 1 MHC in the cells. And so when NSP14 is expressed, class 1 MHC and its partner beta-2 microglobulin are downregulated significantly, but the RNA for all the class 1 A, B, and C alleles are not affected. So this is a protein level change. So then we asked, what kind of chemical perturbation could we use to reverse the downregulation of class 1 MHC? So NSP14 interacts with inosine monophosphate dehydrogenase 2, and there is a classic natural product approved drug inhibitor, mycophenolic acid, that inhibits IMPDH2. And so you see on the right, when we take empty vector expressing cells, mycophenolic acid has modest to no effect on class 1 MHC. But when we express NSP14, the downregulation of class 1 MHC is virtually completely reversed by mycophenolic acid. So this was very, very exciting because we took the protein-protein interaction map, linked it to an existing drug, and found that the phenotype of downregulating class 1 MHC could be reversed by the drug treatment. And you'll notice that the strep tag is reading out NSP14. Mycophenolic acid also leads to a direct reduction in NSP14 level in the cell as well. So that's I'll come back to that in a moment. Now, that was just class 1 on the cell surface, but of course we're trying to get viral peptides to be presented. So Xiang treated cells with NSP14 or empty vector or NSP14 plus mycophenolic acid, immunoprecipitated the class 1 MHC, acid-eluted the peptides and sequenced those peptides. And with empty vector, these are just rough numbers, and we're all aware of the positives, the numbers of detections by mass spec. But I think in general, we got a very nice confirmation that empty vector versus NSP14 dramatically decreased the number of human proteins we detected. When we looked for the peptides from NSP14, we saw 2 under those conditions. But when we added mycophenolic acid, we saw 2 additional peptides that would be detected when we added mycophenolic acid, suggesting we were recapitulating the full presentation pathway. So as I mentioned, NSP14 is a very interesting enzyme. It's a dual enzyme. It's got a 3', 5' exonuclease activity in the N-terminal domain and a methyltransferase that methylates the cap and probably other RNAs in the C-terminal domain. So we individually inactivated each of the enzymatic activities, and very interestingly, when we inactivated the exonuclease, NSP14 was still able to downregulate class 1. But when we inactivated the methyltransferase, we saw an inability of NSP14 to downregulate class 1 MHC. So it seems like the ability to downregulate class 1 MHC is connected with one of the 2 enzyme active sites. Why that is, why methyltransferase might be important for HLA is very interesting. I'd love to hear anybody's ideas about that. We're still puzzling over a pathway that might be responsible for that. But we did think then that we might have a practical way, even if we may not understand how NSP14 downregulates. We know that IMPDH2 interacts with NSP14. That came with the protein-protein interaction map. We recapitulated that interaction in our own cells, and you see in the first lane, IMPDH and NSP14 interact, the flag and the IMPDH2 are bound together, but when we add mycophenolic acid, we disrupt that interaction. So we know that by targeting the host factor, we then have also broken apart that interaction. And I showed you on a previous slide, you see it here too, NSP14 levels are reduced as well. So whether the importance is to just destabilize NSP14 or have an additional effect, we're still working on. And then that's recapitulated by the mutants in the methyltransferase activity. We reduce the interaction and we further reduce it with mycophenolic acid. And now with Adolfo Garcia-Sastry's lab, we're doing full viral infection with mycophenolic acid and seeing what happens during the full infection. And with that, just want to acknowledge my just fantastic postdoc, Xiang Zheng, who really started this project even before I knew he was doing it. And it's great to have such fantastic postdocs in the lab, especially during these pandemic times. So thanks to him, with Adolfo Garcia-Sastry's lab, Chris White, especially Lisa Murin, who's doing the infection assays, and of course, Nevan's lab, David Gordon, Jeffrey Guo, and of course, all the QCRG. Thank you.
Thank you very much. Very interesting. So we have one question from Shaeri, who says, awesome talk. Was wondering if MHC class 1 is degraded or its translation is inhibited. Also, do other viral proteins act differently to inhibit MHC than NSP14?
Yeah, great, Shaeri. We haven't figured out whether it is degradation or lack of production. We're sorting that out right now. And once we figure that out, then we'll be able to compare it to other viruses. But we've never seen a methyltransferase that sort of has this effect on class 1. So we're really intrigued about that.
Maybe a follow-up question. Do you know where NSP14 is localized in the cell?
It seemed in the map that we just have, it's pretty diffuse in the cytoplasm. So it might have some punctate staining in the cytoplasm, but it seems rather broadly expressed.
So there's no chance it's actually at the chromatin level and it could be inhibiting through this methyltransferase activity, the actual expression of the gene.
Right. Maybe I should have clarified. This methyltransferase activity is an RNA methyltransferase. So it's the methylating the cap and probably other viral 5' UTRs to help that fantastically big mRNA translation boost when the virus comes in.
Okay. We have two questions from the Q&A. So one from Suhad Al-Salihi. Thanks for the well-presented talk. On what basis the test chemical mycophenolic acid was selected? Thank you.
Yeah, Sahud. Mycophenolic acid is a natural product discovered in the 19th century, actually. So it's been around for a long time, and it was discovered to be an IMPDH inhibitor. It's the very first one. So it is the prototypical IMPDH inhibitor. There are now more synthetic ones, which we've used as well, but it's sort of the prototype.
Another question from the Q&A from Sam Huang. Is the N7 methyltransferase domain of NSP14 alone sufficient to downregulate MHC1 complex?
Yeah, we should chop it in half. That's a good idea. Is that what you mean? Take one half and just see if that C-terminal half. Like, yeah, I like that idea. Thanks for that suggestion.
Yes, he answered yes.
Yes.
Hey, Kevin, one quick question. Is there work ongoing here to reconstitute this in vitro to test the binding effects in vitro and structurally characterizing this interaction? I didn't know that.
Yeah, Klim is doing that in the structural genomics group. And it's really fascinating because IMPDH has this incredible multimerization property. And when it gets activated or deactivated, it multimerizes and changes shape. So it has 6 nucleotide binding sites to regulate GTP, ATP levels in the cell, and the multimer somehow tunes all of that. So I think mycophenolic acid might break that apart, and I think NSP14 relies on the multimer to somehow sort of ramp up IMPDH activity and not be sensitive to negative regulators and things. So I'm really hoping Klim can do that. It'll be fantastic.
Yeah, cool, very cool.
I have one last question, if no one else has one. I noticed that NSP14 was also interacting with SIR2 and 5, which is a histone deacetylase. So is there any evidence that deacetylation might have an impact on the effect?
Yes, I think actually we're not working on that, but people at the Gladstone are working on that. So it's a great direction. I haven't caught up with Eric and people so at the Buck to find out what the latest is. I think Nevan mentioned that to me recently.
All right. Well, it seems like there is one more question from Philip Alabi. Is the binding site of the receptor of MPA known? And then regarding the receptor, I'll ask the second part afterwards. Go ahead.
Yes, there's a beautiful 1996 Cell paper crystal structure of mycophenolic acid bound adjacent to the nucleotide. So it's one of these active site inhibitors that is non-competitive. It doesn't compete out the substrate. It sits next to the substrate. So it's one of these beautiful natural product mechanisms that only evolution could find.
Complementary question regarding the receptor of MPA. Are there mutagenic studies to observe the effect on presentation of MHC?
Mutagenic, maybe change. I didn't get what.
Maybe mutations in the receptor of MPA that would have an effect on presentation of MHC.
Yeah, not that I'm aware of. I've not seen people link IMPDH to class 1 MHC, so it's kind of the viral, the host, and then this phenotypic screen that led us down this direction.
All right.
Thank you.
Then I think we're done, and we are on time.
We're 1 minute over.
Oh, well, excuse me.
That's very good. I just wanted to say on behalf of my co-organizers, Carmen and Julia and Gerald and Shri, thanks everybody for a great day of science. It was really, really exciting. And we're going to do this again tomorrow, same time, same place. I don't know if anybody else on the Pasteur side wanted to say anything.
No, it was wonderful.
Great job, everybody. Thank you very much.
Thank you. Good night.
Okay.